Hi, myself Priyanka Vijay Kulkarni. In this video, we'll see in detail about the restriction enzymes. What is mean by the restriction enzymes, how they cut, what are the types of the restriction enzyme, all the things we'll see in this video. Yes. So this I already told in my previous video that these restriction enzymes are comes under the class of the nuclease enzyme or the nuclease family. Nuclease means which break down the nucleotides or the nucleic acid or the DNA molecule. So this nuclease enzyme further has the two types, exonuclease and the endonuclease. Exo means outside. Okay, so exonuclease means which cut the outside of the DNA or at the end of the DNA. And the endonuclease means, endo means in between or in middle or the within the DNA. So this restriction enzyme is probably called as a restriction endonucleases. Okay, this enzyme produces the internal cuts which is called as the restriction endonuclease enzyme. This restriction endonuclease enzyme, it is a bacterial origin enzyme. Okay, which is from the bacterial origin, which cuts the double stranded DNA into the fragments. Okay, after the recognition of the restriction site. Yes, first of all, the restriction site or recognition site must be present on the DNA which has to be cut. Okay, so that restriction site is nothing but the one type of the palindromic sequence. Palindromic sequence means a mirror image of that sequence. Yes, so this restriction site is recognized by the restriction enzyme, then it goes and binds to that site and cut that DNA, double stranded DNA molecule. And these are from the bacterial origin, isolated from the different types of the bacteria, and it is evolved by the bacteria to restrict the viral attack. Why it is present in the bacteria? For the self protection of that bacteria. Means if virus attack on that bacteria, then this restriction enzyme will cut the DNA of that viruses. Okay, so for the self protection, these restriction enzymes are present in the different kinds of the bacteria, and it is also known as a molecular Caesar. Yes. Then discovery, how this is discovered or who discovered this. So basically it is originated from the lambda phages. Okay, so there are the actually three types of the restriction enzyme, type 1, type 2 and type 3. So in 1916 by the scientists Arber and the Mendelssohn, they study the type 1 type of the restriction enzyme. Okay, in 1970 the another scientists Hamilton O. Smith and Kelly and the Kate Wilcox, they isolate and characterize the type 2 restriction enzyme, which is the first type 2 restriction enzyme which is invented. It is in 1917. It is a Hind 1 or Hind 2 restriction enzyme. Okay, this is isolated from the Haemophilus influenzae bacteria. Yes, Hind 2, it is a first restriction enzyme, second, which is isolated from the Haemophilus influenzae. And yes, this Nobel Prize in 1978, the Nobel Prize for the psychology of or the medicines was awarded to this scientist for this their work and the discoveries of the restriction enzyme. Yes, there are three types of the restriction enzyme, type 1, type 2 and the type 3. This classification is done on the basis of their composition, the cofactor requirement of this enzyme, the nature of their target sequence, means what are their target sequence or what is the recognition site, okay, and the position of the DNA cleavage site relative to the target sequence. On this basis, these three classes are classified. Yes, so how this restriction endonucleases works? This enzyme, it recognizes the recognition site or the restriction site which is present on the double-stranded DNA molecule, and it produces the double-stranded cut in the DNA, and there are the two types of the cut, blunt end and the sticky end. In this figure we can see what is meant by the blunt end. Blunt end means straight cutting is there, okay, and sticky means there is a overhang, single stranded overhang flanking region is remain. It is known as the sticky ends. Yes, what is this blunt end? This blunt ended fragments, it can be joined to the any other type of the DNA fragments which also has the blunt end, and some enzymes are required for the cloning of such a blunt ended fragment. And the sticky ends, the DNA fragments with the complementary sticky ends can be combined to create a new molecule which allows the creation and manipulation of DNA sequence from the different sources. So basically sticky ends are necessary. It is also known as the staggered cut, okay. Yes, we'll go one by one. Type 1 restriction endonucleases or type 1 restriction enzymes. It is a very complex endonucleases. It has the recognition sequence, okay. This has recognition sequence up to the 15 base pair, and it cleaves the DNA about 1000 base pair away from the 5' end of the sequence TCA located within the recognition site. And the examples of this are ECO K12, ECO B, etc. So basically these restriction enzymes are not more used in this recombinant DNA technology. Type 2 restriction enzymes are probably used, but we'll see what is this type 1 restriction enzyme, okay. This enzyme, it has a particular recognition site within the DNA, so that recognition sequence or that palindromic sequence, it should be the 15 base pair sequence, okay, minimum to maximum 15 base pair sequence. Then after recognition of this 15 base pair sequence, this type 1 restriction enzyme goes and binds to that recognition site or restriction site, and it make a cut to the double stranded DNA, but where it cut the 1000 base pair away from the 5' end of that sequence, means where it is bind, it cut not at that site, it cut 1000 base pair away from the 5' end of that site, okay. And basically in between this 15 base pair or in between this recognition site which is present on the DNA, the probable sequence is a TCA, okay. TCA must be present, and some examples are given here. Then this enzyme works with the help of cofactor, okay. It requires the three types of the cofactor. Which are these cofactor? S-adenosine methionine, okay, S-adomate, this is a one cofactor. Why it is required? For the methylation purpose, yes. Then ATP and the magnesium, these three cofactors are required for the type 1 restriction enzyme. This yes, it requires the adomate, means it is for the methylation. So this enzyme methylate the DNA sequence at the site of recognition, means when it recognize the recognition sequence on that DNA molecule, it goes and binds to the DNA molecule at that restriction or recognition site, and then it methylate that sequence, okay. Then yes, this enzyme translocate the DNA sequence. It also help in the translocation of the DNA sequence. Then this enzyme has the three subunit. Okay, it is made up of from the three different subunit. HSDR, which are required for the restriction or for the actual scissoring purpose, cutting purpose. HSDM, it is necessary for the adding the methyl group. Okay, methyl transferase activity. Then HSDS, it is important for specificity of the cut size recognition in addition to its methyl transferase activity. Okay, it SSDS, it adds the specificity. Okay, yes. Then type 2 restriction enzymes. It is remarkably stable and induces the cleavage either in most cases within or outside their recognition sequence, which are symmetrical. Okay, here also this type 2 restriction enzyme is mostly used in the recombinant DNA technology. This enzyme, it recognizes the recognition or the restriction sequence which is present into the DNA which we have to cut. So it recognizes that sequence and it goes and binds to that sequence. This restriction enzyme binds to that restriction site and then it directly cut at that site. Okay, at between that site or outside that site. Okay, but what about type 1? It cut at the 1000 base pair away from the 5' end of that site, but here it cut within that site or just outside that site. So this is an important thing, and it requires the cofactor. Only one single cofactor is required, that is the magnesium ion. Okay, the first type 2 enzyme is isolated is Hind2. This is a very important, it may ask in a MCQ. Okay, first type 2 enzyme to be isolated was Hind2 in 1970. Only type 2 restriction endonucleases are used in the cloning experiment and the mapping experiment. Why? Because it cleaves at the specific site and type 1 cleaves at the non-specific site. Yes. Then this restriction enzyme, it can generate the two types of the cut depending on whether they cut both the strands at the center of the recognition sequence. Yes, cohesive ends means sticky ends. Okay, or the staggered ends. There are three names: sticky end, staggered end and cohesive ends. Yes, and the blunt ends also. Yes, blunt end means in between. Blunt end means there is a no overhang region, and sticky end means there is a overhang region. Yes, type 3 endonucleases. These are nothing but the intermediate of type 1 and the type 2, and they cleave the DNA in the immediate vicinity of their recognition site. Means they cleave non-specifically but not that much far that of the type 1 restriction enzyme. Okay, they cut the DNA up to 20 to 30 base pair away from its recognition site. Type 2 restriction enzyme is the only enzyme which cut at the recognition site. It is specific. Type 1, it cut 1000 base pair away from the recognition site, and type 3, it cut 20 to 30 base pair away from that recognition site. And it requires the two types of the cofactor, ATP and the magnesium ion. Okay, yes. Then also requires the adomate, means methyl transferase activity, and the ATP cofactor for their role. Yes, here there is a nomenclature is given. A nomenclature is basically given after the name of that bacteria where it is present or where it is from isolated. For example, here three examples are taken: EcoR1, Hind3 and BAM H1. So EcoR1, okay, how the name or nomenclature is given: Eco, ECO, okay, E, capital E, it takes from the genus Escherichia. Okay, E is taken from the genus Escherichia. Then CO taken from its species first two letter, CO. Okay, the strain of this E. coli is R strain, so this EcoR. and one it is a isolated order, okay, or order isolated, it is a one, so the naming is a eco-R1. And the recognition site is also very specific for the each and every restriction enzyme, okay. Each and every restriction enzyme has their particular recognition sequence or recognition site. So here, what is its recognition site on the DNA molecule? GAATTC, and it cuts in between the G and A. Next is the hint 3. Hint 3 means H, capital H, came from the genus Haemophilus. IN, where is IN come? From the species Influenzae, first two letters. D, it is a strain number, and third is the isolate. So this hint 3. Then BAM H1, B from the Bacillus, AM from the Amylo, H it is a strain, and first it is a order, and it cut in between the G, and these are the some recognition sites of this restriction enzymes. Yes, the recognition sequence of this type 2 endonucleases, it form a palindrome with the rotational symmetry. Means whatever the recognition sequence of the type 2 endonuclease, they are nothing but the palindromic sequence of the DNA molecule, okay. They have the recognition site of up to the 4, 5 or 6 base pair, which are palindromic and which is a GC rich. Here are some properties are given, like the nature of the enzyme, then the protein structure, restriction environment, cleavage sites, and the examples. We'll see type 1, type 2 and type 3. Type 1, it shows the endonuclease and methyl transferase activity. Type 2, endonuclease and methyl transferase activity, but it's separated. And type 3, both the activities it shows. Protein structure means it is made up of from the how many subunit? Type 1 made up of from the 3 subunit. Type 2 made up of from the 2 identical subunit, and type 3 made up of from the 2 different subunit. Yes, then the cofactor requirement. Type 1 require the 3 types of the cofactor: ATP, magnesium, and adomate, adenolatic or adenosyl methionine. Yes, then type 2 require only one, yes, only one cofactor, magnesium, and type 3 require ATP and magnesium, 2 cofactor. Cleavage site, yes, type 1 it cut up to the 1000 base pair away from the restriction site at the 5' end. Then it cut very near or in between the restriction site. Type 3, it cut the 10 to 20 or 10 to 30 base pair away from the restriction site, and the examples are also listed over here. Yes, applications of this enzyme, mostly it is used in the gene cloning and the protein expression experiment. Then it is used in the biotechnology to cut the DNA into the smaller fragment in order to study the length of that fragment, okay, by using the RFLP, restriction fragment length polymorphism. Then each of these method depends on the use of the agarose gel electrophoresis for the separation of this DNA molecule. So to separate this DNA fragment, agarose gel electrophoresis technique is used. Then yes, also helpful in the preparation of the recombinant DNA molecule, which has a very wide range of the applications. Then it allows for the large scale production of human insulin for the diabetes using the E. coli, as well as for the hepatitis B and HPV vaccines, also used in the vaccine production or the human protein production, like the cloning DNA molecules, studying nucleotide sequence, all these things. Yes, thank you.